In this video we will discuss about one-step production of optimized CAR. So far whatever the results that we have discussed in this paper, there they produced the optimized CAR in a multi-step process. That is, first they reduce the HLA-ABC overexpression, then they overexpress either PDL1 or this HLA-E, and after that they also optimize this, they transduce this CAR and then check its efficacies. So once they have optimized the conditions, they wanted to see whether they do all the modifications in the immune cells, that is in the NK cells, in a single step or not. So they make a lentivirus that has the CAR to suppress HLA-ABC, and at the same time it also has the CAR construct, and which is the CAR construct that they use is of second generations which have the CD3 zeta and also 4-IBB co-stimulatory domain, and CD3 zeta is for the activations of the NK cells. And also they have this informations about this overexpression of this either PDL1 or SCE, and I have also already said that they have the informations about the suppressing of HLA-ABC. So they did their studies with two different CAR construct, one targeting CD19 and one targeting MSLN antigens, and they check their efficacies, and in the results result shows as per the expectations, that is overexpressions of their MSLN CAR, and at the same time they also have suppression of this HLA-ABC and the overexpression of this PDL1 or the HLA-E, which they transduce using SCE. And here the results only for this MSLN CAR shows, but in the supplemental figures they might have shown the results for the CD19 CAR. So once the results are as per expected, they wanted to see its cytotoxicity effect, whether it has the same efficiencies that they see previously or not. So for that they use their respective cancer cells, that is MSLN CAR is treated with this ovarian cancer cells, and CD19 CAR is treated with this co-cultured with this. CD19 CARs are co-cultured with these Raji cells, and they are able to see that in the top the panel C shows the results for the MSLN and ovarian cancer, and here they can see that when only this HLA-ABC is knocked down, they are able to see some cell lysis. However, this cell lysis will be enhanced with either exogenous expression of PDL1 or exogenous expression of SCE, and they can see the increase till this effector to target ratio 3:1. And they did the similar studies with this CD19 CAR along with the Raji cells, and here they also see that with only HLA-ABC knockdown they are able to see lysis, cancer cell lysis to some extent, but this is cancer cell lysis increased significantly when it is HLA-ABC is knockdown is supplemented with either PDL1 overexpression or SCE overexpressions. So once validating the efficacies of their one-step productions of optimized CAR, they also checked the change in the signaling pathways in the different conditions. For that they used RNA-seq analysis of this different kind of NK cells, and what they are able to see that when only the CAR NK cells are used without any modifications of HLA or PD1 or HLA-E, they are able to see that the cell stress pathways are overexpressed, and these stress pathways could be because of the activations of these NK cells once they detect their respective cancer cells. And these things they have seen for both they have seen for the NK cells for both MSLN and also for the CD19. Here the right, and thereafter when they check the results with the check the results of this CAR NK along with this exogenous, either exogenous PD-L1 or exogenous SE overexpressions. They are able to see that those cell stress pathways are suppressed, and at the same time the cell cytotoxicity pathways are upregulated. For example, IF and gamma signalings, and also IF and gamma signaling, and also there's granzymes signaling pathways are upregulated. So this indicates that cell cytotoxic pathways are upregulated, and which is what they have expected based on the results that they have seen previously. And at the same time, the interesting thing that they're able to see that suppression of the cell stress, indicating that it will increase the life of the NK CAR cells. So this is what they have found out, that this is what the results shows about the one-step production optimized CAR, that it not only has almost similar efficacy that they see in the multi-step CAR optimization method, and at the same time when they check their signaling pathways or changes that occurs within their CAR construct at different conditions, those changes are also in line with their biological those changes also make the biological sense. At the same time, it also gives indications of the long life of this CAR NK cells because of the low stress observed by them. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.